Hello friends, welcome back to my YouTube channel Nasrin Padasale. So in this video we are going to discuss the answers for those questions that has been asked in code number 1 agriculture biotechnology. In case if you wrote your examination in some other question paper, please do share your questions in my mail ID on the comment section of this particular video. So I'll be prepared the answers for those questions also. So in this video we are going to discuss only for agriculture biotechnology. First question is regarding to what are the component of transcription unit. So transcription unit generally consists of promoter, coding region and terminator. Next question is regarding to DNA transfer technology, that to specifically microprojectile method, which is also called as particle bombardment or biolistic method. They are asked about the initial velocity. Okay, so micro projectile is a method which is generally employed for DNA delivery of your DNA to a plant cell. Okay, so it's a generally most commonly employed method. So they will be employing a heavy microparticle which might be a tungsten or gold, where they will be coating the microparticle with your DNA of interest, and it is accelerated to a initial velocity of nearly 1400 feet per second. So the correct answer, the initial velocity is 1400. If you want, you can able to check in their website. You can able to find a lot of information about microprojectile and about some other DNA transfer technique. So next question is regarding to what is the effect of charcoal? Why charcoal is added to plant tissue culture? So generally they will be adding as an activated charcoal which will be trapping toxin and inhibitory component. Okay, so by adding these charcoal to your plant tissue culture media, it will be preventing the absorption of these toxic compound by a plant. Okay, and next important toxic inhibitory compound that are produced by plant tissue culture in plant tissue culture is generally phenolic compound. So this adding of this activated charcoal generally reduce the accumulation of your oxidized phenolic compound. Okay, next thing, a Sanger's reagent is used for identification of N-terminal end of your protein. So it is generally used in protein sequencing in order to determine the N-terminal amino acid. So Sanger's reagent is also called as DNFE, that is fluorodinitrobenzene. Okay, so this particular DNFE, they will be undergoing few kind of nucleophilic aromatic substitution with the N-terminal amino acid group of your protein or a peptide group. Okay, so then after hydrolysis of your peptide bond, the individual amino acid will be separated, and there will be this particular reagent will be labeling only N terminal of your amino acid side chain. Next is overlapping gene produces how many proteins? So overlapping genes means what? It is a dual coding genes. They are the regions of DNA, or it might be a region of RNA that are translated in two different reading frames. Each will be having a separate promoter and separate terminator. Due to few overlapping circumstances, they are getting overlap. Okay, so the question is that how many proteins are produced? Okay, so this particular overlapping genes, since they are translated into two different reading frames, they will be yielding two different proteins. Next is CPD enzyme is related to. So what is meant by CPD? It is constitutive photomorphogenesis dwarfism, and this particular enzyme is specifically involved in your brassinosteroid biosynthesis. Okay. Next is which ethylene receptor lack histidine kinase domain. Okay, so if you take, there are ethylene receptor is classified into two subfamily. One is called as ETR1 and ERS1, which have a canonical histidine kinase domain. Whereas if you take this ETR2, ERS2, EIN4, they are belonging to a subfamily 2 of ethylene receptor, and they have consist of a diverged histidine kinase domain, and they also lack this particular histidine kinase activity. Okay, they have a diverged histidine kinase domain which don't have the histidine kinase activity. The correct answer is EIN4. Next is what are the important component of binary vector? Binary vector will be consisting of origin of replication of E. coli, origin of replication for agrobacterium, so that it is acting as a shuttle vector. Next is consist of multiple cloning site, plant selectable marker and bacterial selectable marker. In addition to that, it also have a promoter region, reporter region, and poly signal. Next, which population is used for marker assisted selection in plant breeding? So generally for marker assisted selection, they will be employing a near isogenic line population, recombinant inbreed line population, double haploid and backcross, as well as they will be employing F2 population. Okay. So next, what is the definition of biofortification? So biofortification is nothing but it's a process where they will be improving the nutritional quality of your food crops. One such example is golden rice. Okay. They will be employing either few agronomic practices or conventional breeding, or few biotechnology based approaches also will be employed for this biofortification. Next, when molecular beacon will be showing fluorescence in PCR, the correct answer is during the annealing step. So what does mean by molecular beacon? Means they are generally employs a employ real-time PCR, so they are single-stranded DNA molecule. They are generally having hairpin shaped, and they are oligonucleotide probes which are employed for your real-time PCR technique. Okay, so this hairpin, one end will be having a fluorophore and another one will be having a quencher compound. Okay, so in the presence of target sequence, this molecular pigment will be unfold and they will be bind to a target sequence. So in that case, fluorophore and quencher will be separated. So fluorescence will be detected at the annealing step. The fluorescence will be detected. Okay, so in each annealing step, the fluorescence will be monitored and reported. Okay. Next is what is meant by split gene, which split gene is also called as interrupted gene. The particular gene is defined as a consisting of an intron, which is also called as intervening sequences between exon, and it also has a exon segments of interrupted gene that are represented in mRNA. So simply the split gene will be having at least 2 exon followed by 1 intron between the 2 exon. Next is inhibitor binds to enzyme substrate complex alone is called as, it is called as uncompetitive inhibition. Next, what is the purpose of E2 hybrid system? Okay, so generally this E2 hybrid system is generally employed for both for your protein-protein interaction studies and protein-DNA interaction studies. Next is a neurotransmitter which is found in animal and also in plant. So the particular compound will be acting as a neurotransmitter in animal, whereas in plant it will be acting as an important signal for growth and development. So I don't know complete answer, but correct answer is tryptophan, because if you take tryptophan, it is acting as a basic precursor molecule for producing various kind of neurotransmitters. In the plant, it will be acting as a precursor for the biosynthesis of various plant hormones like your auxin. Next, the 3D structure of microtubule will be dimeric in nature. Next question is, which of the following is not a direct method of gene transfer? The correct answer for this question is vector mediated gene transfer, which is also called as agrobacterium mediated gene transfer, because this is called as indirect gene transfer technique, because they will be employing few TA plasmid vector for transferring a DNA into the plant cell. Okay, whereas some other direct gene transfer technique include your microprojector method, and some other methods will be employed where they will be directly injecting the DNA of interest into the plant cell. They won't employ any kind of vector. Next, the Taq polymerase. What is the nature of Taq polymerase? So Taq polymerase is called as thermostable DNA polymerase 1. There are 3 DNA polymerase. DNA polymerase 1, 2, 3. The Taq polymerase is belonging to DNA polymerase 1 group, and the Taq polymerase don't have any 3' to 5' proofreading activity. So that's why in these recent days they are not employing Taq polymerase enzyme for the PCR reaction. They are employing some other thermostable DNA polymerases. Okay. Next is regarding to enhancer. First, enhancer, they are non-coding region and they are non-coding regulatory region present in your gene, and they will be either located upstream of a gene or they will be located in downstream of the gene. At the same time, they will be mediating both temporal specific transcription control and tissue specific transcription control. They also employ long distance control with the promoter region. Next is urease is the first crystalline enzyme which was crystallized by the James and Robert. Okay, both group of persons are involved in identifying this particular urease. Okay, and they, after identifying urease only, they had identified enzymes or proteins. Like that, they had came to that particular conclusion. Next, the enzyme which is used in Nick translation is called as DNase 1. Okay, next I have listed over macronutrient, micronutrient. So you can able to find the correct answer. Macronutrient include carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, sulfur, calcium, magnesium, and silicon. Whereas micronutrient include other compounds like iron, manganese, copper, zinc, molybdenum, boron, chloride, nickel, sodium, and other things. Okay, cobalt. Next, according to Jacob and Mort's equation, repressor will be binding to operator. I think this time from lac operon 2 question has been asked. One, which enzyme is involved in lac operon? It is beta galactosidase. Next question is regarding to repressor. In which particular region repressor bind? Repressor will be binding to an operator region. Next, they are asked which particular RNA polymerase is involved in transcribing tRNA. I think so. So here you can able to find the easy answer for. At the same time, you can able to use this particular slide for your future preparation. So RNA pol 3 is involved in transcribing a transfer RNA. Okay, whereas RNA polymerase 2 is involved in transcribing your mRNA, and RNA pol 1 is involved in transcribing your ribosomal RNA. Next is regarding to ETC inhibitor. So antimycin A is an antibiotic which will be inhibiting the electron transport chain by disturbing complex 1 structure. So it will be binding to the quinol site of cytochrome C reductase, and it will be inhibiting the oxidation of your ubiquinol. Okay, whereas cyanide, it will be inhibiting the electron transport chain by binding to the complex number 4. Okay, Next is regarding to molecular marker. The dominant marker include RAPD, AFLP, EST, ISR, whereas codominant marker include RFLP, SEAR, SSR, SMP, and STS. Next, only in prokaryotic transcription. So, if you take the transcription and they had asked the question which particular organism, both the transcription and translation will be occurring simultaneously. The correct answer is prokaryote. Okay. So, in prokaryote, both your translation and transcription will be occurring simultaneously, but this is not impossible in eukaryotes. We all know transcription will be occurring in membrane bound nucleus, where after the transcription mRNA will be exported to the cytoplasm, and thus in cytoplasm only a protein species will be occur. So that's why in eukaryote both the translation and transcription are not coupled, whereas in prokaryote both the processes are coupled. Next is with regard to colchicine, which is an alkaloid which is derived from that particular plant and generally inhibit the mitosis at metaphase stage. This colchicine will be inhibiting microtubule polymerization. You should know some drugs will be inhibiting the mitosis by inhibiting the microtubule depolymerization, but this colchicine will be inhibiting your mitosis at metaphase stage by inhibiting this microtubule polymerization. Next is regarding to teichoic acid. Teichoic acid is found in the cell wall of gram-positive bacteria, and these teichoic acid are generally anionic polymer which is made up of phosphoglycerol phosphate unit. Polyglycerol phosphate unit. Next, there is a question regarding to succinate dehydrogenase. So, succinate dehydrogenase is an enzyme which will be converting your succinate to fumarate, and this succinate dehydrogenase is an important enzyme which will be linking your Krebs cycle with the oxidative phosphorylation. So, next there is a question: what is the function of glycerol in DNA loading? Right? So, glycerol will be acting as a density substance. So, it will be equivalent to your honey. So, it will be keep your DNA inside the well. So, once you have loaded your DNA into the well, it should be kept inside the well. So, glycerol will be adding density to your DNA molecule. Next is immunogold labeling technique, which is also called as immunogold staining technique, which will be employed, which is a unique immunostaining technique, which will be employing your electron microscopy and generally employed for detecting your protein, antigen, and other macromolecule within the location of cell itself. Okay. Next is what is meant by Km. So Km is the concentration of substrate at which half of the active site of enzymes are occupied by that particular substrate. Okay. So this Km value is the concentration of substrate which half of the Vmax is attained, and this Km also determines the binding capacity or affinity of enzyme towards a particular substances. Next, which gene is employed? Which gene of baseless thuringiensis is used in BT cotton? It is Cry1AC gene is employed. Next is regarding to ionocetal, which is also called as myoinocetal, dichiro-ionocetal, and hexaphosphate. They play a critical role in body cellular growth. Okay, so this ionocetal generally referred as vitamin B8, but this particular compound is not actually a vitamin. It is a type of sugar molecule that helps your body to process the insulin. Next, which of the following is not a PCR based molecular marker? It is RFLP. So RFLP is a one such molecular marker which doesn't employ PCR. Instead of that, they will be employing a restriction digestion technique. So it will be compare an individual based on the polymorphisms in their genome. Next is a single nucleotide polymorphism. So single nucleotide polymorphism means this polymorphism will be caused by point mutation, which will be giving different allele consisting of alternative bases at given position of nucleotide within a locus. So this single nucleotide polymorphism are generally biallelic in nature. Okay, so you can refer this particular article. Next is histone modification. So histone modification generally regulates your replication coupled nucleosome assembly. Next, it will be regulating your DNA damage repair. At the same time, it also plays a significant role in gene transcription. So histone modification play a role in replication as well as transcription. Next, male sterility gene include your Barnase gene. This Barnase gene expression will be producing an enzyme which will be destroying the tapetal layer that's surrounding the pollen sac. So as a result, pollen will not be formed. It will be leading to male sterility. Next, what are the staining type? Protein will be stained by cum-acid brilliant blue. Nucleic acid will be stained by epidemium bromine, and lipids will be stained by Sudan. Next is EcoR1. The sequence of EcoR1 restriction enzyme is GAATTC. Next is common gall disease is caused by Agrobacterium tumefaciens, which is a gram-negative bacilloform bacterium which is associated with the roots. Next is Yak vector. So Yak vector, size of Yak vector will be ranging from 1000 kb up to 3000 kb. Okay, so general the size will be ranging from 1200 kb. I don't think so. I don't have the option with me, so I'm giving you a value in between them. Okay, so ACDC transposable control element was the first transposable element system which is recognized in your maize. Next is how many ATP and NADH is produced in glycolysis. So glycolysis, you all know it will be starting with one molecule of glucose which will be producing 2 molecule of pyruvate. So total amount of ATP is 4 ATPs are needed and 2 molecules of NADH are consumed to convert the glucose to pyruvate. Next is alternate splicing. Alternative splicing will be expanding a proteome complexity, which will be generally generating a multiple transcript or multiple protein isoform from a single gene. Next is TBB, that is Tata binding protein will be complex with 14 TAF, that is Tata binding protein associative factor, to form the general transcription factor called TF2D. Okay. Next, they are asked in which part of ribosome does the 16S rRNA will be residing. So it will be residing in a prokaryotic smaller subunit, which is also called as 30S subunit of your 70S ribosome. Okay. So this 30S subunit will be holding the 16S ribosomal RNA, and additionally it also holds 19 protein molecule. Next is most of the eukaryotic cellular molecule, cellular mRNA are modified at their 5' end by the addition of this particular group called 7-methyl guanosine. Okay, they form a specific triphosphate linkage called 5' to 5' triphosphate linkage, which will be connecting this particular cap to the molecule. So this is a few kind of post-transcriptional processing that will be occurring only in the eukaryotic mRNA. Next, there is a question regarding to cell cycling checkpoint. So there are 3 checkpoints will be there. So first checkpoint is G1, which will be occurring at G1 to S phase transition, which will be checking whether the cell is in correct stage to undergo for cell division. Okay, and next checkpoint will be G2 checkpoint, which will be occurring in between G2 to M phase transition, and it will be accessing whether the DNA is completely duplicated at S phase or not. Okay, and the last checkpoint will be occurring in the mitosis stage. Okay, and the transition from mitosis to anaphase, there is a checkpoint called spindle assembly checkpoints will be there. Okay, so this spindle assembly checkpoint will be ensuring that chromosomes will be properly attached or not, whether the chromosomes are properly aligned to the spindle. Okay, so these are the 3 important checkpoints. So that I don't what is the correct question, but after the AM phase, the next checkpoint will be activating on G1 phase. Okay, next ribosome, which is also called as ribonucleic acid. These are RNA molecule that have a catalytic activity, which will be catalyzing few specific biochemical reaction like RNA splicing. Next, northern blot is employed for studying RNA, whereas southern blot is for DNA and western blot is for protein. So nucleoid is nothing but it's a nucleus like they are irregularly shaped region which is present in prokaryotic cell, and that will be holding nearly all genetic material of that particular prokaryotic cell. Next, what is meant by prosthetic group? So first, prosthetic group is a non-amino acid compound. It might be either your vitamins or few ionic compound that will be forming a structure with a heteroprotein. Okay, so protein without this prosthetic group is called as apoprotein. So when this apoprotein they binds with the prosthetic group, it is called as holoprotein. Next, in eukaryote, primary RNA transcript is called as either they will be calling as a primary RNA transcript or it will be calling as hnRNA, which is also called as heterogeneous nuclear RNA. Okay, so this heterogeneous nuclear RNA will be going through few kind of modification to produce as a mature RNA. This mature RNA will be transported out from the nucleus. Next is what is meant by hybrid. Hybrid is a hybrid of cells or plant which will be consisting of the genome of one species but cytoplasm of both the species. That's why they are called as hybrid. Okay, next is interspecific hybrids. So interspecific hybrids are generally produced by a technique called embryo rescue, where they will be collecting those embryo which are not having any capacity to form a mature plant. Okay, so they will be collecting the embryo and they will be growing them in the lab. Okay. Next is with regarding to GM mustard, there is a question has been asked. So this GM mustard was developed by Professor Deepak from University of Delhi. Okay, so the main aim of producing this particular mustard is that they need to reduce the India demand for edible oil import. Okay, so that's a major aim for this regarding to mustard. So there totally 3 genes are applied. One is bar gene, next is barnase gene, next is bar star gene. So barnase will be conferring a male sterility, just I had explained you previously. Bar star will be restoring the ability to produce fertile seeds. Next is called as bar gene. So this bar gene is responsible for glufosinate resistant. Okay, so they will be producing the enzyme called phosphinothiosin N-acetyl transferase. So this enzyme is responsible for providing resistance against this particular phenolic compound. Okay, next is BACMAP system will be using a modified insect cell virus called baculovirus, and this BACMAP system is generally employed for delivering or expressing genes in mammalian cell with minimum effort at the same time minimum toxicity. Next, which protein are involved in polar transport of your auxin? It is spin protein. Another protein is called as AVCB protein, which will be acting as an auxin efflux carrier. Next, with regard to two-dimensional gel electrophoresis, which will be employing IEF, which is called as isoelectric focusing. Next is SDS, which both the technique are employed. That's why they are called as 2D gel electrophoresis. Next is SAGE, which is called as serial analysis of gene expression, which will be employing a mRNA from a particular sample and they will be creating a complementary cDNA fragment. Okay, so SAGE is for producing cDNA from mRNA. So what is meant by comparative genomics? So comparative genomics is the field where you will be directly comparing a complete genetic material of one organism against the other organism in order to gain a better understanding about how a particular species have been evolved, and how they are also helpful in determining the function of genes and non-coding regions in your genome. So next is regarding the energy source with regarding DNA ligase. So ligase produced from T4, that is T4 ligase will be employing ATP, whereas E. coli DNA ligase enzyme will be employing NAD. Next, who discovered the first enzyme? So he was the one, he was a German physiologist called Kuhn. He was born in Hamburg and he was the one who had coined the term called enzyme. Next, glycosylation occurs. Glycosylation occurs in majority of glycosylation occurs in Golgi. So mainly a O-link glycosylation will be occurring in Golgi, whereas at N-link glycosylation, half will be occurring in ER and half will be occurring in Golgi. Next, who discovered genetic code? Okay, so the Marshall Nierenberg along with Robert Hawley and Hal Gobin Corona were the one who had discovered the genetic code, and for which they had got a Nobel Prize in the year 1968. Regarding to Francis Crick, he was the one who gave the codon concept. Okay, so I don't know about the correct question because few are telling Francis Crick is the correct answer, but for me, the correct answer is Marshall Nierenberg and Argobin and Robert Hawley. Next is this regarding to fusicoin, which is a ditryphenyl glycoside which is produced by this particular fungus, and this fungus is generally acting as a parasite in your almond tree and peach tree. And the main function of this particular glycoside is that they will be stimulating a acidification of plants. How come? They will be activating a H plus ATPase pump, and this particular acidification will cause the stomata to get open all the time, which will be ultimately make the water to get into the cell, and it will be producing a death of that particular plant. Next, permeability is stopped due to accumulation of this particular compound called suberin in crop. Okay, so suberins are generally found in peridot, which is nothing but your crop layer, that is the outermost layer of your bark, and this layer is generally dead only, and they have abundant amount of this particular compound called suberin. So this suberin, they generally prevent the water loss from the tissues that are present below the cord. Next, what is the receptor for jasmonate? So jasmonate, the receptor is COI1 is the receptor for jasmonic acid signaling in plant. Next, who was the one who had identified golden rice? So golden rice was given by this particular Portuguese who was a professor in Swiss Federal Institute of Technology, and Peter Bayer, who was a professor in this particular University of Edinburgh, Germany, and both belong to Rockefeller Foundation Initiative. They were the one who had discovered this golden rice, but the inventor has partnered with this particular company called Syngenta. Okay, so currently Syngenta is manufacturing, and they are involved and they are engaged in activities related to golden rice. Next is Tata bugs. So Tata bugs are generally located 25 to 35 base pair upstream of the transcription start step. Okay.